Thank you. Thank you, Professor Hato, for the nice introduction. Thanks for inviting me here, and thanks for coming to my seminar. It's been my great honor to be here talking to you about my research on mechanics and geometry in chiral structures, from helical seed pods to twisted embryonic brain. So I'm going to talk to you about some of my work as a PhD student at Princeton and some of my postdoctoral work. So here's the outline. First, I will give you a very brief introduction of linear elasticity theory, and then for the main part, we're going to talk about tunable helical ribbons and embryonic brain torsion before giving the concluding remarks. First, the introduction. So to appreciate this talk, all you need now is love. The first two L refers to Landau Lifshitz, who wrote this very nice little book, Theory of Elasticity. And for those of you who are diligent and smart, you should dive into the book by Love, treatise on the mathematical theory of elasticity. And really, to appreciate today's talk, what you need is just a little bit knowledge of linear elasticity theory. So given an initially undeformed round body, if under certain mechanical loads it deforms into, say, the shape of a heart, then a point here moves to a point there, and if we simply do the subtraction, that gives us the components of the displacement vector. And if we just do the following differentiation, that gives us a quantity called strain tensor. So these are the components of the strain tensor that quantifies how much a body is strained in 3 dimensions. And in linear elasticity theory, people just throw out the higher order terms, and the constitutive law simply states that the stresses are linearly proportional to strains, and that's like a 3D analogy of the well-known Hooke's law. So force is linearly proportional to displacement in 1D. Now let's move on to the first topic, tunable helical ribbons. We study helical structures because helices are almost everywhere in nature, going from DNA double helix to alpha helix, lipid bilayers, bacteria flagella, to macroscopic objects, horns of animals, tendrils, and seed pods of plants. And engineers have also been interested in making nanohelices that can be used as sensors, resonators, transistors, and biomedical devices. So the goal of our study is to develop strategies for designing 3-dimensional helical ribbons on demand. And for that, we would need a theoretical description of 3D helical ribbon deformation. And traditionally, there have been these one-dimensional models that treat helices as just one-dimensional line objects in space. And for those of you who study solid mechanics, you're probably familiar with Stoney's or Timoshenko formula for beam deformation. But those cannot handle the general 3-dimensional helical ribbon deformation. So the challenge here is, given an initially flat, straight ribbon, what kind of magic can you perform to obtain a spontaneously deformed helical ribbon? So I'm going to pass along the helical ribbons that I made a few years ago. So you can see they're relatively, they stay in shape for 3-4 years. And graduate students, you can think about how you're going to make these helical shapes if your supervisor asks you to work on this. And so the easiest thing one can think of to make a helical ribbon is you take the initially straight ribbon and you just bend it around a cylinder. Let's say a cylinder with a horizontal axis with a radius of curvature 1 over kappa 1. In this case, you get a left-handed helix. Or alternatively, you can bend it along a vertical cylinder. And in this case, you can get a right-handed helical ribbon with presumably a different radius of curvature. But in so doing, we notice that the edges of these helical ribbons will have to be straight because you're simply bounding them on a cylinder. Nevertheless, the helical ribbon shapes observed in natural systems, such as in lipid bilayers, can have non-straight edges. So the edges can, in fact, be either concave or convex. So the question really becomes, what happens if this ribbon wants to bend both along the horizontal and vertical axis, and presumably with different radius of curvature? How would these two competing processes compromise? So where this end configuration become a left-handed or right-handed shape. So if now we zoom in such a deformed helical ribbon, we can see that this little patch here actually deforms into kind of a cap shape with two principal curvatures, kappa 1 and kappa 2, along the principal bending directions R1 and R2. And in general, these principal bending directions do not have to line up with the geometric axis, so the length and width direction of the ribbon. And so there could be a misorientation angle phi between these two axes. And so the question becomes, if we know these three independent geometric parameters, kappa 1, kappa 2, and phi, how do we describe the 3D configuration of this ribbon in space? It turns out to be a non-trivial problem, but we can solve it using differential geometry, and you can look at the details in our papers. But towards the end of the day, the center line of the ribbon can be parameterized as a function of the arc length measured from the beginning. And all these other parameters can be written in terms of kappa 1, kappa 2, and phi, these three independent geometric parameters. And we can show that this is actually, the center line actually forms a helix, and the macroscopic geometric parameters. So the helix angle can be written in terms of kappa 1, kappa 2, and phi. And the handedness or chirality is simply dictated by the sign of the helix angle. And the pitch is the distance between the adjacent terms. And the radius of the bounding cylinder can all be written in terms of these three independent geometric parameters. Now, if we plot them, so we can see that by altering the relative values of kappa 1 and kappa 2 and phi. So here we actually fix the value of phi to be 45 degrees and just change the relative values of kappa 1 and kappa 2. We can get a bunch of different shapes. So we can have cylindrical helical shape with straight edges when one of the principal curvature is 0. And in general, when they are both non-zero, we can have a general helical ribbon with either concave or convex edges. And this is a particular case when the principal curvatures have different signs. And in fact, it becomes a purely twisted ribbon when kappa 1 equals minus kappa 2, and the angle is 45 degrees. And when kappa 1 equals kappa 2, the ribbon would form a ring shape. And that's the geometry part of the problem. And so the next question becomes, how do we get these geometric parameters, kappa 1, kappa 2, and phi, from the material parameters, the Young's modulus, Poisson's ratio, etc. So for that, we resort to solid mechanics. So in this model, we put surface stresses as the driving force, because in a lot of helical structures observed in natural and engineering systems, they're down to the micro and nano sizes where the surface to volume ratio becomes very significant. And so the surface stresses are tensors. They can have these principal directions, so E1 and E2, plus minus just to know the surface stresses on the top and bottom layer, respectively. And we assume for the ribbon geometry that the thickness is much smaller than width, and width is much smaller than length. So the strain tensor that characterizes the deformation are the following. These white ones are the in-plane strain components, and the green ones are the strain components related to bending, because when you bend a ribbon, so there are parts that's being compressed and there are parts that's being stretched. So the strain here will be linearly proportional to z, which is the distance away from the neutral plane. And there could also be these residual strains. Now, if we plug them in this formula, we can calculate the total energy associated with this bending. So these are the contributions from the surface stresses, which tends to lower the energy, whereas the red part is the elastic strain energy. So there are penalties in elastically deforming the body. And we know nature favors the lowest energy configuration, so we do energy minimization with respect to the unknown parameters to find out these geometric parameters of interest. And for a particular problem, we can actually have analytic solutions. So if there's only one surface stress on, let's say, on the bottom surface, then we can calculate these geometric parameters from the driving forces and material parameters. And we can show that in the absence of Poisson's effect, our solutions actually can be reduced to the classical Stoney's formula. And so the Stoney's formula is really one specific example of ours. Of course, if we only have theory, that's not so fun. So we perform these tabletop experiments that you have seen. So the way we design these experiments are the following. So we take a sheet of latex rubber and we can pre-stretch it along perpendicular directions. Okay, so we can tune how much it's pre-stretched in these two directions, and then we bond a tape along a tunable angle. So this bonded two-layer system, upon release, would spontaneously deform into one of the helical shapes that you might have seen that we're passing along. So here I'm showing you the comparison between experiments, the white ones, and the theoretical predictions. So we can see that when the misorientation angle is 0 and 90 degrees, we have ring structures. And that's because the helix angle becomes 0 when either phi becomes 0 or 90 degrees. Okay? And otherwise, we get helical ribbons with varying pitch distance and changing radius of the bounding cylinder when the driving forces are anisotropic, so the pre-stretches in two directions are different. But what happens when the pre-stretches in these two directions are the same? Can anybody guess? Okay. Any guess?
What was the question?
So the question is, when the pre-stretches in these two different, these perpendicular directions are the same, what kind of shape are you going to get? So I'm showing you here when the pre-stretches in two directions are different, then we have these tunable helical ribbons, right? Then what happens when, the pre-stretches are the same? Do we still get these helical shapes? Great, yeah. So that's what we get. We get rings regardless of the misorientation angle. And that can be interpreted from our theory because the helix angle now becomes 0 since kappa 1 equals kappa 2. Okay, so regardless of the misorientation angle, we get 0 helix angle, which reduces it to a ring. Okay, and we can further make a purely twisted shape, and that's by a 3-layer experiment in which we pre-stretch the top and bottom layer in perpendicular directions. So that would dictate that kappa 1 equals minus kappa 2, okay, because it wants to bend in this shape. And we sandwich a strip in between at the 45 degrees angle. And if you do the calculation, the radius of the bounding cylinder becomes 0, and it becomes a purely twisted shape. Okay? And we can further show that transitions between these different shapes, so purely twisted shape or cylindrical helical shape with straight edges, and general helical shapes with either convex or concave edges. And a closed tubular structure can be made by just tuning these relevant geometric parameters. Now we also tune the width because in some biological systems, the width of the ribbon can also grow. And I'm going to briefly mention about our work on bistable structures. And this is actually inspired by a toy. So some of you might have played with this. So it can be stable in this very straight configuration. And it's made of stainless steel, very stiff. But if you tap it on your wrist, it will quickly transition into a coiled shape. So it can be stable in more than one shape. This is bistable, and it has 2 stable states. I'm going to also pass it along so you can play with it. Oops. And we can actually manufacture such structure by a 3-layer experiment. So we pre-stretch the top and bottom layer with the same string. And we sandwich a square sheet in between, so they're bonded together and let go. It's not surprising that the system will deform into a saddle shape because of the nature of the driving forces. Okay? But what is interesting is if we have exactly the same driving forces, but we just change the dimensions, we make the strip wide enough and thin enough, then interesting things starts to happen. So the system now, instead of being stable in just one saddle shape, it can be stable in 2 shapes. And you can apply a mechanical stimulus to make it transition from one shape to the other, pretty much like the slap bracelet. Okay. So why? The driving forces are exactly the same. We just change the width or the thickness. We have a theory which I'm not presenting in full here, but from our theory we can calculate the bending energy and the stretching energy associated with this large deformation, and then we can compare them and find that this eta is a control parameter that would dictate the loss or gain of bistability. It's related to the width, the thickness, and the curvature. And the reason being that the stretching energy over bending energy goes as eta to the 4th. And so when eta exceeds a threshold, meaning when width becomes very large or when it becomes very thin, the stretching energy will be dominating, which is not good because we know nature always likes lowest energy configurations. But it's okay if the system adopts a shape with nearly 0 Gauss curvature. So this is the product of the principal curvature is a Gauss curvature. So even though this term may be dominating, if the Gauss curvature is nearly vanishing, it's still fine. So that's why the system when eta is very large, we would prefer to bifurcate into one of these nearly cylindrical shapes instead of being in a saddle shape. And that's the geometric condition for bistability. And there's a very nice paper that was almost published concurrently with our work that deals with the helical formation of the seed pods. And they also designed these tunable helical shapes. And they also proposed a similar parameter to ours that dictates a bistability. And we can compare their experimental results of the threshold value of eta to our theoretical prediction, and we find very good agreement. And further, we noticed that this kappa involved in this dimensionless parameter is actually unknown to begin with. So we think this might not be the best parameter for us to design bistable structures because we really don't know kappa to begin with. So we propose an equivalent dimensionless parameter which we can show. So everything here is known. So we have the driving force, we have Young's modulus, thickness, width, and we can use this parameter to design bistable morphine structures. And there's a colleague from Hong Kong who recently contacted me about what they discovered at the nanoscale. So they found that strained semiconductors can also have this kind of multi-stable behavior, and which they didn't notice before, and I pointed out to him is actually similar to what we've been studying. So they propose to use this as micro robotic manipulators, like grippers. And now, if we simply combine these two studies together, so before we have these tunable helical shapes, and so what happens if we just increase the width given exactly the same driving forces? In this case, we found that instead of having helical ribbons with varying radius, now we have helical shapes with almost the same radius of bounding cylinder. And they all oriented along the direction where the driving force is bigger. And that's, again, dictated by the competition between bending and stretching energy. The system now prefers to stay in a nearly cylindrical shape, okay? And it's monostable. More interesting things can happen. For example, when we simply increase the dimensions for this case, so we manufacture the purely twisted shapes before. So if we, again, keep exactly the same driving forces and we just change the dimensions, can you guess what's going to happen? Because of the competition between bending and stretching energy, we talk about this. So, any guess? Okay, so first of all, like in this case, we know when it becomes very wide, so eta exceeds the threshold, the system is going to adopt kind of a nearly cylindrical shape, right? And that's what happens, okay? So this guy becomes nearly cylindrical, given exactly the same driving forces. Moreover, it can actually adopt another helical shape with a different orientation. And so it becomes actually bistable helices. And I can show you a video, so that's like this one. So it can go from a shape with almost 30 degrees helix angle to a shape with a 60 degrees helix angle. And it can go back and forth. Okay. And the shape transition from a purely twisted shape to nearly cylindrical shapes can actually occur in a variety of physical systems. For example, in liquid crystals, when there's a change of temperature, such transition can occur. And we've recently collaborated with a group of chemists and chemical engineer at Emory University and Georgia Tech, the David Lin group and Martha Grover. So we studied the transition from a purely twisted shape to nearly cylindrical helical ribbon and to closed tubular shapes in peptides. And we found that just by changing the width, it actually such shape transition can occur, and the theoretical prediction agrees reasonably well with the experimental data. So to sum up for the first part, we found that surface stresses can induce helical morphologies, and we proposed a theoretical model that gives predictions beyond the Stoney's or Timoshenko formula. And we showed that mechanical anisotropy and geometric misorientation together can induce helicity. And the sources of mechanical anisotropy can come from anisotropy in surface stresses, residual strains, or elastic modulus. And we show that smooth transitions between or these different shapes can occur when we tune the parameters, and that if we have control over the morphology via external stimuli, so that would give us ways of designing sensors, transducers, or drug delivery systems. And we also show that geometric nonlinearity can result in bistability. And putting these together, we give the principles of shape selection in helical ribbons and with associated instability. So now let's move on to talk about the mechanics of morphogenesis in embryonic brain torsion. So in the past century, the scientific community has embraced the huge success of molecular and genetic studies of development, but it remains poorly understood how the one-dimensional information coded in DNA is translated through mechanics into three-dimensional biological forms, a process termed morphogenesis. And you can see that these embryos look pretty similar in shape in terms of their shapes early on, but later they can develop into dramatically different organisms. So here we choose to study the brain morphogenesis because such studies can benefit our understanding of congenital brain defects and hopefully can benefit practices in preventing such defects. The brain defects occur in almost 1% of live human births. And it's been known that abnormalities in size and shape of the developing brain would cause the serious neurological disorders, such as anencephaly, which involves missing part of the brain, and when there's abnormal swelling of the brain, hydrocephalus can occur. Other neurological disorders include autism, schizophrenia, and mental retardation. And in our lab, we use chick embryos as a model system because the early development of chick embryos goes almost parallel to that in the humans. For example, the first 3 days in chick development goes parallel to the first 3 weeks in humans. Here we study the early brain development, so we use a fertilized egg. We can incubate it to any desirable stage, and we use a filter paper to carry it onto a petri dish and observe its development under the microscope. So after about 36 hours of incubation, so you can see that the brain tube is relatively straight, and later it starts to subdivide into forebrain, midbrain, and hindbrain, and then it starts to expand and bend. It would bend ventrally, and then it starts to twist. And so it basically turns 90 degrees such that the left side of it would lie on the yolk. So here's a movie showing you this process. And we're looking at this embryo from the ventral side. You can see the heart is beating, and the brain is going to bend towards us eventually, and then it starts to twist, and it almost always twists rightward. So if you imagine I'm an embryo, I'm going to bend like that, and I'm going to twist rightward. And this is one of the earliest organ-level left-right asymmetry events in development. And it's known that inverse rotation can be associated with birth defects like situs inversus, when the internal organs are in the mirror image positions of the normal ones. And so this process is also termed axial rotation. And so if we look at it from the ventral side, it eventually becomes a question mark. And so here are the questions we're trying to address. What forces cause the brain to twist? What determines the direction of twist? And does bending affect torsion?
Can I ask you a question?
Sure.
I don't think of the brain as being a long, skinny thing. I think of
Okay.
it as being like this. So
Right.
what part of it are we looking at?
So we're looking at this early brain. So you can see this part is the forebrain, and this is the midbrain, this is the hindbrain. So later on, I can show you, probably in another slide. So this is an early stage, about 3 days of development that we've been studying. But later, it's gonna undergo dramatic shape transformations into the brain models that you often see.
I mean, do we see this twisting in brains as they get older, or is this just a temporary twist when they're very small?
This is a temporary twist, right? But this is a process, like in chick embryos and mouse embryos. It has to undergo this process to assume normal development. Okay. So normally, as we can see, that the brain would twist rightward, but occasionally we found it twists to the opposite direction. But we found in both cases the heart resides on the concave position. So, for example, in this case, heart is on the right-hand side when the brain turns rightward. And in that case, the heart is on the left-hand side when the brain turns leftward. So is there any connections between the direction of the heart looping and the direction of brain twisting? If we look at the anatomy, so we use optical coherent tomography to see this. So there's actually a membrane that's on top of the embryo that's called the Vittering membrane. And this is the heart. And there's another membrane that's right beneath the heart that's called splenomegaly, or SPL. And the brain bends. The bending of the brain is also termed flexure. There are several flexures. So in between forebrain and midbrain, there's cranial flexure. And around the hindbrain and the spinal cord, there's cervical flexure. And in the spinal cord, there's another flexure which actually has a different sign of curvature in the opposite direction to those two, which we'll call thoracic flexure. And so our hypotheses are the following. So we hypothesized that the Vittering membrane that's on top of the embryo actually constrains the brain flexure, thus causing the brain to twist. Okay? And second, that the direction of the looping heart would actually determine the direction of the brain torsion. And third, the degree of flexure would affect brain torsion. To test our hypothesis, we performed the following experiments. So first, since we think the Vittering membrane is exerting a force necessary for torsion, so we removed the Vittering membrane at around 40 hours of incubation. And so after 17 hours of incubation, we found that the embryonic brain torsion is much less than in a control embryo. Okay? And if we further cultivate it for 11 hours or so, the brain torsion increases, but still is dramatically smaller than the torsion in a control embryo where it twists almost 90 degrees. And we can actually apply an alternative force that can cause the brain to twist immediately. And that's through the fluid surface tension. So, and I'm going to show you the experiment momentarily. So these results support our hypothesis that vitreous membrane is necessary for normal brain torsion. So here's the movie. You can see the heart is beating. So we simply lower the level of the culture media, and you can see that by applying the surface tension, the embryo immediately twists to a position that's comparable to a control embryo. And that's in real time.
Question. It looks like it tends to go torsion to protect the heart. Is that the direction it goes?
Right. Yeah. And for our second hypothesis, since we think the direction of the heart looping would affect the direction of brain twisting, so we designed experiments in which we push the heart from the right side. So this is the ventral view, from the right side to the left. Okay, and then we culture it for about 12 hours, and we found that the brain actually twists leftward in comparison to that in a normal chick embryo. And so that supports our hypothesis that it is because of the asymmetric position of the heart that there's a preferred twisting direction of the brain. And we also observed that if the brain already has significant amount of torsion, then even if we push the heart to the left side, the direction of brain twisting stays the same. And this is actually because if we examine it closely enough, so we found that when the brain already has significant torsion, the hard push to the left side will return to the right side in minutes. And so, in that case, when we culture the embryo, the twisting of the brain is about the same as in a control embryo because the heart cannot stay on the left side. And we also build finite atom models using a simplified geometry of the embryo. So these are the flexures, and that's the heart that loops to the right side. So that's the brain. And we apply a force from the top that represents the force from the vitreous membrane. And that's the SPL membrane beneath the heart. And we can take a look at the results. So the force from the vitreous membrane can actually cause the brain to twist rightward. That's the head-on view. And we can see that if we remove the heart from our model, apparently the brain is just going to bend. Whereas when the heart loops to the right side, the brain would twist rightward. And if we tune the distance between the heart and the SPL membrane, you can see that the brain undergoes just simple bending before the heart touches the SPL, and then it starts to twist rightward. And we also build a physical model through 3D printing from our computational model. And then we can use a cover slip to apply a force that represents the force from the Vitreous membrane. And here we show the comparison between the deformed shape of a physical model and the shape in a real embryo. And that's under moderate surface tension. And if we further apply the surface tension, then the embryo is going to twist completely. And we can also measure the torsional angle associated in these two processes. So the blue ones are the torsional angles that's involved in a real embryo, and that's averaged over 4 embryos. And here I'm showing you how we measure the torsional angle in a 3D printed physical model. And so we plug these needles on the side of the physical model, and then we apply the surface tension by using a cover slip, and then we can measure the angle between, the change of angle of these needles, and that's represented here, the red ones. So the trends in these two models are similar. Last but not least, we investigate the relationship between the bending and twisting of the brain. To do this, we implant a eyelash into the brain tube to suppress the bending part. And so you can see that when the bending is suppressed, the torsion is also suppressed. Okay, so now, for example, you can see the two eyes in comparison to what happens in the control embryo, where it turns almost 90 degrees. And we also noticed if the eyelash did not reach the first pair of somites, for example, then the suppression of the cranial flexure does not prevent or does not suppress the brain from twisting. So it still twists almost 90 degrees, indicating that suppression of the cervical and thoracic flexures are also important for brain torsion. So just by suppressing the first flexure does not induce a reduction of brain torsion. So to sum up, we showed that the Vitreous membrane applies an external force that are necessary for the brain to twist in a chick embryo, and that the direction of heart looping actually determines the direction of brain torsion. And that's why, in most cases, the brain twists rightward. That's because, in most cases, the heart loops to the right. And that the suppression of the flexure, and in particular the second and third flexure, would reduce the brain torsion. So to conclude, I hope I've convinced you that by studying the relationship of mechanics and geometry, we're studying morphogenesis, which can bring us endless fun. And that's because there are endless problems we can solve. And in particular, I show you that the combination of mechanical anisotropy and geometric misorientation can give rise to helical geometries, and that geometric nonlinearity would induce bistability. And putting them together, we can come up with shape selection principles of helical structures and the associated instability. And for the brain torsion problem, we showed that external forces from the Vitreous membrane is important in causing the brain to twist, and that the asymmetry of the heart looping actually determines the direction of the brain torsion. And I'd like to also share with you a quote about our methodology. This is from an anonymous lab. So theory and practice. Theory is when you know everything but nothing works. Okay. Practice is when everything works but nobody knows why. In our lab, theory and practice are combined. Okay. Nothing works, and nobody knows why. Okay, that was a joke, but I hope in my future group, okay, we strive to understand how things work in nature, and then we use the ideas, the principles learned from nature, we build things that work. With that, I'd like to acknowledge the support from my PhD supervisors, Dr. Mikko Hadaya, David Strauss, and my postdoc supervisor, Dr. Larry Tabor, and my colleagues that have contributed, some of them contributed to our work, and my faculty mentors and the lab mates. And I'd also like to acknowledge the funding sources, in particular the Society in Science Branko-Weiss Fellowship for the generous support. With that, I'd like to thank you very much for your attention, and I'd be happy to address your questions. Sure.
Question about the metastable shapes.
Mm-hmm.
So most of them seem to be like they were in two different states of coiling. Although the thing that you passed around was straight, and then it would coil.
Right.
But
it's actually not straight. I mean,
exactly.
it's straight in one position, one direction, but it's curved in the other. Right.
And are there any practical issues associated with taking something that would be in a metastable state curled and with very little force would straighten out or go linear? I mean, I'm just wondering about the sort of the mechanics of these structures if they really want to be coiled in some way.
Right. I actually have one such thing. I'm not sure whether it's exactly what we're talking about. So it's like a magic bar. So it can be very short, okay? But you press a button, and it suddenly goes very long. Is that what you're
Basically
talking about?
Like that. Something that's compact.
Right.
I mean, I have applications in mind.
Right.
In space, for example.
Okay. Yeah, I probably could have brought that toy and maybe I can show you sometime. Yeah. Okay.
I'm wondering how you make, I understand how you make your stretched latex models.
Right.
How is the bracelet made?
Oh, you're talking about the way we make it, or?
The snap bracelet.
Oh, this one.
How is that made?
Oh, so I think the idea here is also induce kind of plastic deformation in two directions, such that locally it has tendencies to bend in this direction, and it has tendency to bend in the other direction. And then, because of these geometric nonlinear effects we talk about, it has to adopt kind of a nearly cylindrical shape. So it has to choose one shape or the other. And so, in fact, if you cut the snap bracelet down to a small enough piece, then it loses this bistability, and it becomes just a saddle shape.
So, so you say it's a plastic deformation. How is that manufactured?
Oh, I think they use a thermal method. So, for example, you have a mold that's cylindrical shape, and then you conform it to the surface, and let it undergo some thermal treatment so that it's stable in this direction. It becomes a cylindrical shape in this direction. And same thing for the other direction. Okay. Sure.
I have two questions.
Yep.
Is a fiddling membrane always in compression? You say the heart moves because the membrane.
Oh, actually, I think it's actually in tension. So it's like a membrane. So it's in tension, and so it will apply a force.
You're right. The surface forces are concerned.
Right.
Medically speaking, would it be possible to put some sort of drug into the membrane so that it doesn't change? It doesn't put stresses upon the embryo.
To put some what on the membrane?
Is it possible to somehow affect the membrane so that it relaxes and does not?
Oh. That's a great question.
That's just something to think about. But
Right,
I have
right.
another question.
Yeah.
Could you go back to the slide that you showed of the equation that fitted data to?
Which, are you talking about brain torsion
You
or?
had a physical model. And
Oh, this one.
The human
Is it this one?
on one slide.
Is it this slide?
You're showing data. Yes, that one.
Okay.
Well, looking at the red data,
Right.
I would say you haven't fitted the curve correctly.
Mm-hmm.
Looks to me as if there's a translational relationship about 2/3 up.
About two.
And if you move that back, you get one equation.
Which sorry, which?
In the middle. You see where the horizontal line? The red data.
This part?
It's at 20.
20.
On the horizontal line?
Oh, this.
Yeah, right there.
Right.
There's a translation.
Mm-hmm. Yeah, right.
So that your curve, I think, is one curve, and then there's a translation in the middle.
Right, right.
Which
So,
tells you something about the blue ones.
mm-hmm. Right. So I think this fitting is kind of, I mean, it's not the best. I mean, we just simply use a like an arrow function
You've got
to
another
fit it.
relationship to look for right there.
Right. I agree. This could be an important feature of this deformation. Right. Yeah. Thanks. Mm-hmm.
So I have a question.
Right.
You're looking at brain twisting at this stage of embryonic development to the next stage, and showed that it's associated with asymmetry. But somewhere before that, the asymmetry in the heart had to develop. Any idea of what happens before you get to the?
Before the heart.
I mean, I guess you start with 2 cells and you eventually get a heart, a beating heart.
Right.
It's like that due to some asymmetry causes the brain to twist.
Right.
Any idea what causes that initial asymmetry?
You mean why the heart goes to the right side?
Yeah.
Yeah. So actually, the Tabor lab has studied this problem before, and from the study, I think what happens is there's 2 veins. So initially the heart is a straight tube too, and then it starts to bend into a C shape, and then it starts to twist, and it goes to one side. And it is shown that the forces from these 2 veins are unequal, and also that this membrane, the SPL, the Spanish nuclear membrane, applies a load that causes the heart to go one way or another. And in fact, in our experiments, so let's see. So here we were able to keep the heart on the left side because we still have the SPL membrane here, on top of the heart. Or alternatively, if we lower the fluid level, we can use the surface tension to keep the heart on one side. Otherwise, the heart would beat and then it would slowly go back to its original position. Yeah. So we've done a series of experiments to test this. And so when the heart is kept on the left side, then the brain twists in an opposite direction. If there's no membrane to keep the heart on the left side, then the heart will return to the right-hand side. Then the normal twisting direction we'll assume.
So if this
Sure.
were in a human, how old would the human embryo be to be at this stage?
This is comparable to about around 2 days. Oh no, sorry, 2 weeks. Yeah. So the first 3 days goes parallel almost to the first 3 weeks.
So could this be seen by ultrasound?
Actually, I think so. The interesting thing about brain torsion is that it's kind of species dependent. So what we studied today, I think it's generally true for species with a big yolk. So, for example, all the avians, chick, duck, other species, birds, basically, and some, I think, alligators or like species with large yolk will have this kind of
Large what?
yolk.
Yolk.
Right. So the twisting would be 90 degrees. Okay. But in mouse embryos, interestingly, or rodents, the twisting is 180 degrees. I think I've shown that in yesterday's talk.
Well, do we expect
And-
any of this to be relevant to humans?
Right. So I think in humans, it's kind of controversial right now because I've looked into this issue. So I've seen pictures that show rightward torsion in some kind of rightward torsion in human embryos in a comparable stage, but I'm actually not completely sure about whether that mechanism is the same as what happens in chick embryos, and definitely I think that would be different from the mouse embryos. And that also raises interesting question from the evolutionary point of view, like how do these mechanisms, evolve or preserve, between species? Sure.
Can you put a dimension on that? 2 centimeters?
Dimensions? Oh, yeah. So this is like 1 millimeter.
1
The
millimeter?
millimeter, yeah.
So it's
the
about
bar.
2 millimeters wide.
Yeah,
Right.
yeah, close to that.
Thank you.
Yeah, no problem.
Okay. Does this have any relevance to the effect of ethanol on the process?
To the effect of ethanol?
The phenomena that you're talking about, the surface relationships and forces might be affected by ethanol.
Oh, you mean chemical?
Alcohol.
Yeah. Well, we don't have alcohols there. Also,
There's fetal alcohol syndrome, and I assume that's what you're talking about.
alcohol syndrome?
The mother consumes alcohol.
Oh, if the mother consumes alcohol. Oh, wow.
I can make a leap here.
Right. There's a leaf there.
So I guess the
Yeah.
question being, might chemicals disturb the
Right. It's not impossible, but I wouldn't assume it would have a direct. I don't know that.
But it might be an interesting avenue.
Yeah, it might be an interesting issue. Yeah, right. So how would the nutrients or chemical have an effect on development?
We're dealing with a process of gestation.
Right.
Where this phenomenon could be
Right.
very early in the process before
Right.
anyone is aware that there's embryo present.
Right. Maybe it'd be interesting if we put some droplets of alcohol and see what it does. When the chick embryos get drunk.
Just one more question. I know
Oh.
I asked about ultrasounds, looking at human embryos.
Uh-huh.
Ultrasound, is that a possibility? To see this?
It may be possible, but I think the thing is, at this stage, the human embryos is very small. And, I've seen some embryonic, embryology pictures of human embryos at this stage, but I think they are SEM images, so they're taken out. Maybe some imaging people can answer this question better, or people from School of Medicine.
If there aren't any more questions, I will ask one that has much practical applications.
Okay.
Do you see how to apply what you know about twisting to fixing my phone cord?
Tangling. Right. That's actually related to what I talked about yesterday, the perversions, right?
Yeah.
So there would be, in some cases, the change of...
That idea. Maybe you should consult with the phone company.
Right.
Thank you very much.
Thanks. Thank you for your attention.